I want you to try and visualize something for a second. Imagine you have this unprecedented access to the absolute earliest microscopic data from the very first known COVID-19 patients.
Right, the absolute ground zero data.
Exactly. You are staring at the foundational genetic sequences, like the literal building blocks of a virus that is about to completely upend the world as we know it.
Yeah.
And you're looking for the raw, messy, natural architecture of a novel pathogen making its clumsy leap from an animal into a human. But as you comb through the billions of lines of genetic code, you don't just see nature. You stumble across something that makes your blood run cold.
Oh, wow.
Woven directly into this early data, you find the genetic equivalent of a made-in-a-laboratory barcode.
I mean, it would be the ultimate needle in a genomic haystack. And finding something like that wouldn't just be an anomaly. It would completely shatter the foundational expectation of what you were actually looking at.
Yeah, it challenges the fundamental origin story of a global crisis. It's an image that just stops you in your tracks. And that staggering claim, this idea of finding artificial, man-made genetic fingerprints buried inside the earliest patient data, is the bedrock of our deep dive today.
It's a huge topic.
It is. We are pulling apart a very extensive, incredibly dense interview from a Dutch YouTube channel called Lighthouse TV. The host, Flavio, sits down for a long-form conversation with Dr. Rogier Louwen, a molecular biologist who brings forward a data set that is going to demand a lot of unpacking.
And it's a data set and an analysis that is highly technical, but more than that, it is deeply disruptive to the established narrative we've all been living with for years. I mean, Louwen isn't just pointing out a curiosity, he is raising an alarm that goes to the very core of modern diagnostic science.
Okay, let's unpack this. The mission for this deep dive is to rigorously explore Louwen's complex claims. We are going to look at his specific assertion that he located artificial synthetic lab elements inside public data sets that originated from Wuhan. And crucially, we have to explore the massive cascading implications of the two main hypotheses he presents to explain how those elements got there. Because either we are currently living through a catastrophic crisis of global lab contamination, or
we're dealing with a circulating engineered virus.
Exactly, which is a terrifying fork in the road. Both of those pathways carry profound, almost existential implications for the future of molecular biology, how we diagnose disease, and how we manage global public health.
Absolutely.
Now, before we go a single step further, we need to pause for a crucial neutrality check. I am speaking directly to you, the listener, right now. The source material we're examining today contains highly politically charged content.
It really does.
It features intense, unsparing criticisms of the established scientific community, major national public health figures, and the WHO. It dives deep into extremely controversial theories regarding the origins of the pandemic and questions the safety of widespread medical interventions.
And we should be completely upfront here. The claims Louwen makes run directly counter to the consensus held by the vast majority of major international health organizations. This is not mainstream consensus science.
Right. So we need to be crystal clear about our role here. We are not here to take sides. We are not here to endorse these theories, nor are we here to independently validate or verify the scientific claims made by Dr. Louwen in this interview.
We are just the guides.
Exactly. Our job as your guides is strictly to impartially report on what is being discussed in this specific source material. We're going to break down the highly complex science he references, explain the underlying logic of his arguments, and map out the debate so that you can actually understand the architecture of his claims. We are exploring the source, not verifying it.
Our focus is purely on the how and the why. When he throws out complex terms from molecular biology, we want to translate those mechanics so you can see exactly how he arrives at his conclusions, regardless of whether you agree with them.
Let's get into it. Because before we can even begin to make sense of the incredibly dense genetic data Louwen claims to be holding, we have to understand the man holding the data.
That context is everything.
Right. We need to understand his background and, maybe more importantly, why a guy with his resume is currently operating entirely outside of the traditional scientific establishment.
Yeah, when evaluating the weight of a claim, you really have to trace the trajectory of the scientist making it. And if you listen to the interview, it becomes immediately apparent that Dr. Rogier Louwen is not some guy operating out of his garage who suddenly decided to weigh in on genetics.
No, definitely no.
He is someone with a deeply entrenched 25-year career within high-level institutional science.
Louwen details a background that includes a stint at the highly prestigious Scripps Research Institute over in San Diego, and then a massive 22-year tenure at the Erasmus Medical Center, the Erasmus MC in the Netherlands. For anyone who doesn't know, Erasmus MC isn't a small-town clinic. It is a major, heavy-hitting European research hospital. It's the big leagues.
It absolutely is. And his foundational work during those decades was not in obscure theoretical fringe areas. He was working at the absolute cutting edge of gene therapy and CRISPR-Cas technology.
Right.
The interview spends a lot of time up front establishing his technical bona fides because it's vital to the story he's about to tell. For instance, he goes into deep detail about a highly advanced, incredibly specific experiment he spearheaded involving adenoviral and lentiviral vectors.
Okay, let's hit the brakes there, because the interview throws those terms around like everyone has a PhD in genetics. If I'm trying to picture this, what is an adenoviral vector, and what is he actually doing with it?
Think of it this way. A virus, by its very nature, is essentially a microscopic delivery system. Its entire evolutionary purpose is to latch onto a cell, breach the outer wall, and dump its own genetic payload inside to hijack the cell's machinery.
Right, it's like a home invader with a flash drive full of bad instructions.
Exactly. So in gene therapy, scientists realized they could use that natural mechanism to their advantage. To create a vector, they take an adenovirus, which normally causes something like a common cold, and they literally hollow it out.
They just empty it.
Yeah, they strip out the dangerous genetic code that makes you sick, and they replace it with a specific, healthy piece of DNA that a patient might be missing.
So it's like hijacking the delivery truck. You take out the explosives, pack the back with medical supplies, but you let the truck keep its GPS and its keys so it can still drive right into the target cell.
That's a really great way to conceptualize it. And Louwen's work was utilizing this exact truck delivery system to try and cure a severe genetic disorder in rats called Crigler-Najjar syndrome.
What happens to a rat with that syndrome?
It's a brutal condition. In Crigler-Najjar, the animal, or human, as this affects people too, has a faulty piece of DNA that prevents their body from properly breaking down bilirubin, which is a waste product in the blood. Because this waste builds up uncontrollably, the subject experiences severe, life-threatening jaundice. The rats literally turn a vibrant yellow.
That sounds awful.
It is. So Louwen's experiment involved creating one of these hollowed-out viral vectors, packing it with the correct functioning gene that breaks down bilirubin, and injecting it into the rats. The virus acts as the delivery vehicle, infecting the cells, but instead of making the rats sick with a cold, it permanently installs the corrected genetic code.
And Louwen claims that to prove this worked, they visually demonstrated the results. The jaundiced yellow rat pups, after being injected with this genetic delivery truck, physically turned stark white. They completely cured the genetic defect.
Which is undeniable, tangible proof of highly advanced genetic manipulation skills. You don't pull off an experiment like that without intimately knowing how to cut, paste, deliver, and track DNA at a molecular level. He is an absolute expert in the specific microscopic tools used to modify genetics.
Which obviously becomes the entire crux of the story later when he starts claiming to see evidence of genetic modification in public datasets. His background provides the exact lens required to spot these anomalies.
Exactly.
The interview also notes he transitioned into microbiology, studying how bacteria evade our immune systems, and he even secured a major European Union grant for a project called Stamina, which was focused on developing diagnostics for potential epidemics using CRISPR technology.
Actually, let's unpack CRISPR for a moment, because it's another term that gets tossed around constantly.
Good point. How does CRISPR fit into his tool belt? I know it's often called genetic scissors.
That's the popular shorthand, but mechanically it's fascinating. CRISPR is actually a natural defense mechanism that bacteria use to fight off viruses. When a virus attacks a bacterium, the bacterium can capture a tiny snippet of the virus's DNA and save it like a mugshot in a database.
Oh, that's clever.
Right. If that same virus attacks again, the bacterium uses that mugshot to guide an enzyme, usually one called Cas9, to go find the matching viral DNA and literally slice it to pieces, destroying the virus.
It's a bacterial immune system.
Precisely. And what scientists like Louwen learned to do was program those genetic scissors. They realized they could swap out the mugshot for any sequence of DNA they wanted. You can program CRISPR to find a specific sequence in a human genome, cut it, and paste in something new. Louwen was developing ways to use this incredibly precise targeting system to diagnose new epidemics.
No, he is undeniably a seasoned, highly decorated veteran of molecular biology. He knows how the machinery works. But listening to the interview, the tone shifts so sharply from his past achievements to this incredibly dramatic, bitter exit from the institution. Because despite this 22-year tenure, Louwen details that he was fired, seemingly out of nowhere, in 2022.
The official reason put forward by the Erasmus MC, as stated during the conversation, was a refusal to work. But Louwen and Flavio construct a very different narrative. They frame his dismissal not as an HR issue, but as direct, punitive retaliation for his highly critical stance on the hospital's COVID-19 policies and his open rebellion against the broader scientific narrative of the time.
It paints a picture of this massive, irreconcilable schism. I want to break down the three main areas of conflict the interview highlights because you can really feel the alienation he experienced. It starts, fundamentally, with his refusal to take the mRNA vaccine.
Right. Louwen details that he was approached by the Occupational Health Service at the hospital to get vaccinated, which was standard for employees. He outright refused. And he's very clear that his refusal wasn't based on a gut feeling. It was based on his own extensive literature review.
Yeah, he points out that his decades of background in developing diagnostics and his deep familiarity with the scientific literature made him extremely skeptical.
He specifically cites historical failures in developing vaccines for other RNA viruses like SARS-1, MERS, and RSV.
This is a crucial piece of his argument. He claims in the interview that when scientists previously tried to create vaccines for these specific respiratory RNA viruses, the animal trials resulted in disastrous complications.
Yes.
Why? What actually happened in those old trials? Why did they fail?
It's a phenomenon often referred to as antibody-dependent enhancement, or ADE. In some of these historical trials, like early attempts at an RSV vaccine in the 1960s or SARS-1 vaccines in the early 2000s, the vaccine would seemingly work. It would generate antibodies.
Okay, that sounds like a success.
But when the vaccinated animals were later exposed to the wild virus out in the world, the antibodies didn't neutralize the virus. Instead, they acted like a Trojan horse, actually helping the virus enter the animal cells more efficiently.
That is horrifying. So the vaccine made the actual infection worse.
Dramatically worse. The immune system would violently overreact, leading to severe lung inflammation. In many of these historical animal trials, the animals became extremely sick and died upon natural exposure. Because Louwen was hyper-aware of this specific historical context regarding RNA viruses, he felt the rapid global rollout of this entirely new mRNA technology was premature, reckless, and inherently unsafe.
And here is where the institutional conflict really starts to boil over. According to Louwen, his medical privacy was fundamentally breached. He claims that his refusal to vaccinate, which was a confidential medical decision given directly to the Occupational Health Service, didn't stay confidential. He alleges it was leaked directly to senior members of the outbreak management team, the OMT, who operated out of Erasmus University.
To understand the stakes here, the OMT in the Netherlands wasn't just a hospital committee. It was the primary, high-level advisory body to the national government during the pandemic. They were the ones driving national policy. The interview mentions figures like Diederik Gommers, a highly prominent ICU boss, and a leading OMT member. Louwen suggests that his private medical choice was inappropriately shared with these powerful figures, which led to him being summoned for a highly uncomfortable meeting. He says he felt intensely pressured and basically realized in that room that his days at the institution were numbered.
It sounds like a total catastrophic breakdown of trust between the individual scientist and the towering institution. But the vaccine mandate wasn't his only point of friction. The interview spends a huge amount of time focusing on his severe critique of the PCR test, specifically his attack on what they called the Drosten paper.
This is a really vital element of his broader conflict with the establishment. The Drosten paper, which was co-authored by Marion Koopmans, another highly influential figure at Erasmus MC and a key advisor to the WHO, was the foundational defining protocol used globally for the PCR testing of SARS-CoV-2. It set the standard for how the world would diagnose the disease.
And Flavio and Louwen point out something about this paper that made me literally pause the video. They claim that this highly complex paper, which established the global testing standard for billions of people, was peer-reviewed in just 24 hours.
Yeah, that's their claim.
Now, I really struggle to accept that at face value. How does a 24-hour peer review for a complex molecular biology paper even work mechanically? Is that even physically possible?
Well, you're hitting on the exact point Louwen makes. In the traditional, rigorous scientific publishing process, Peer review is notoriously slow and methodical. When a scientist submits a manuscript, the journal editors have to go find independent experts in that specific, highly niche field who have no conflict of interest.
Right, people who actually understand the exact thing being written about.
Exactly. Those experts then have to actually find the time to carefully read the methodology, scrutinize the raw data, check the controls, and ensure the conclusions are genuinely supported by the evidence. This process normally takes weeks, sometimes many months.
Right, because you have to let the information digest. An expert has to sit there and think, wait, did they run a control group for this specific enzyme? What happens if this primer interacts with this weird secondary chemical? You can't just skim a paper that is going to dictate global health policy while drinking your morning coffee.
Exactly. Louwen argues forcefully that a 24-hour turnaround for a foundational diagnostic paper of this magnitude is practically impossible if a genuinely thorough, rigorous, independent review is being conducted. He strongly implies the process was rubber-stamped.
And he didn't just critique the bureaucratic timeline of the publication?
No, he claims he and his team at Erasmus actually took the Drosten protocol and rigorously tested it themselves in the lab.
And what did his team find when they ran the protocol?
According to Louwen, they found that the gold standard test had a massive, dangerous error margin. He claims the specific PCR protocol outlined in the Drosten paper could frequently yield false positives. It was reacting to other viruses, to random background bacteria, or even to the genetic material of the human host being swabbed.
Wait, let me make sure I understand the mechanics of PCR, because this is important. A PCR test is supposed to amplify a very specific tiny sequence of the target virus's DNA, right? It uses these things called primers that act like search dogs. They sniff through the genetic noise, looking for a highly specific scent.
Yes, that's a perfect analogy. The primers are short sequences of DNA synthesized in a lab. They are designed to be the exact complementary match to a specific target sequence on the virus. They act like a chemical lock and key. Once those search dogs find the matching sequence and latch on, the PCR machine uses temperature cycles to copy that sequence over and over, amplifying it exponentially until there is enough of it to be easily detectable.
So Louwen is basically claiming that the search dogs designed in the Drosten paper were poorly trained. That instead of only latching on to SARS-CoV-2, they were accidentally latching onto the wrong sense, maybe a harmless bacteria living in your throat, a different seasonal cold virus, or even fragments of your own epithelial cells.
Yep.
And then the machine dutifully amplified that wrong scent, leading a doctor to tell a healthy person they were positive for COVID.
That is exactly his assertion. Mechanically, if a primer is 20 base pairs long, but 18 of those pairs accidentally match a sequence found in a common human bacteria, the primer might still bind tightly enough to trigger the amplification process.
Wow.
Louwen argues that rolling out a test with such a wide error margin on a global scale, especially to test vast populations of entirely asymptomatic people, was scientifically reckless. He voiced these concerns internally at Erasmus MC, which, according to the narrative in the interview, only deepened the rift and painted a target on his back.
Which brings us to the third part of this institutional conflict, the actual culture within the hospital itself. To back up Louwen's claims of being targeted, the interview references an investigation by a major Dutch news program called Nieuwsuur.
Yes. The Nieuwsuur report, as it's described in the video, investigated allegations of a severe, pervasive culture of fear, specifically at the Erasmus MC Intensive Care Unit, under the leadership of the former boss, Diederik Gommers, the same man on the OMT we mentioned earlier.
The report alleged that critical doctors who dared to speak up about patient workload, safety risks, or disagreements with protocols were systematically intimidated, isolated, or even banished to what some staff darkly referred to as a punishment camp. They were literally physically restricted from entering certain parts of their own department.
Now, it's important to clarify that Louwen was a molecular biologist, not a doctor in the ICU department. However, the interview uses this high-profile news report to validate and contextualize Louwen's personal experience.
Right.
He argues that this overarching culture of silencing dissent and punishing whistleblowers permeated the entire institution. When he spoke up about the PCR tests and the vaccines, he says his scientific integrity was formally called into question by his employer as part of the justification for his dismissal.
But Flavio makes a massive triumphant point of noting something that happened after Louwen was fired. He brings up the LOEI, which is the National Scientific Integrity Board in the Netherlands. Louwen was investigated by them, and they completely cleared him of any scientific integrity violations whatsoever. In fact, the implication is that he was basically fired for being almost too meticulous about data transparency.
It creates a very powerful, compelling narrative arc for the listener right at the beginning of the interview. Here's the brilliant, meticulous, highly experienced scientist who is ruthlessly punished by a dogmatic, frightened institution simply for asking inconvenient questions. But then he is ultimately vindicated by an independent national ethics board.
It's quite the setup.
It is. This framing is absolutely essential to establish before the interview reveals his new, explosive discovery. It tells the audience this man is credible and he has nothing left to lose.
Right. Because having established this dramatic, contentious exit, the video explains that Louwen didn't just pack up and fade into obscurity, he went rogue. He set up his own independent laboratory in a town called Olvenhout. He is operating entirely outside the institutional system now. And from this independent lab, looking at data on his own terms, he makes the core claim of this entire broadcast. He claims to have found something terrifying hidden in the data.
He shifted his focus entirely to analyzing publicly available genetic datasets. Specifically, he downloaded the sequencing data of lung fluid taken from 5 of the absolute earliest known COVID-19 patients in Wuhan, China, dating all the way back to late 2019 and early 2020.
This is the raw data, the actual unedited genetic code of whatever was inside the lungs of the very first victims before the virus spread globally. And Louwen claims that when he ran his own independent bioinformatics analysis on this code, he found what he calls artificial elements. Things that, according to him, simply do not exist in nature, have no evolutionary reason to be there, and must have originated from a laboratory setting.
This is where we need to slow down and do a real deep dive into the molecular biology, because the terms he uses are highly specific. And to understand why he is so alarmed, you have to understand what these tools actually do.
Right.
He identifies 3 distinct artificial elements in these public data sets. The first one he flags is what he calls a neomycin resistance cassette.
Okay, neomycin resistance cassette. What in the world is that? And more importantly, why is Louwen saying it has absolutely no business floating around in the lung fluid of a patient suffering from a novel respiratory virus in Wuhan?
To understand why it's such a red flag, we have to understand the physical reality of how genetic engineering works in a laboratory. Let's say you are a researcher and you want to insert a brand new modified gene into a batch of cells in a petri dish. You introduce the new genetic material, but you immediately face a practical problem.
What's the problem?
Out of the millions of cells in that dish, how do you know which ones actually successfully absorbed your new gene and which ones completely ignored it? You can't just peer through a microscope and see the new DNA.
Right, they don't exactly raise their hands and say, I got the upgrade.
Exactly.
Yeah.
So, for decades, scientists have used a very clever, somewhat brutal trick. When they build the genetic package they want to insert, they don't just include the target gene. They attach a second gene right next to it, a resistance cassette. This specific cassette is a piece of code that grants the cell immunity, or resistance, to a specific antibiotic. In this case, the antibiotic is neomycin, or a specific variant of it called G418.
Oh, I see where this is going. It's a survival test.
It is a microscopic survival of the fittest. You introduce your genetic package to the dish of cells, then you flood the entire dish with the toxic antibiotic, neomycin. Now, G418 works by blocking protein synthesis in eukaryotic cells. It stops them from functioning. So every single cell that failed to take up your genetic package simply dies.
That is brutal.
The only cells that survive the toxic flood are the ones that successfully integrated the resistance cassette, and therefore, because they are attached, also successfully integrated your target gene.
It is a brutal sorting mechanism. It is a classic fundamental tool of laboratory selection designed by humans to force a desired outcome. It is a mechanism of artificial selection, not natural evolution. So finding the genetic code for that specific cassette just casually sitting inside the raw sequencing data of a wild respiratory virus is insane.
Correct. It is a man-made construct engineered specifically for in vitro, meaning in the glass and the lab experiments. Finding it mixed into raw, in vivo patient data is, as Louwen argues, a glaring flashing anomaly.
Wow.
But that was only the first element he claims to have found. The second element is a sequence called the SV40 promoter enhancer.
Now, Flavio notes during the interview that this SV40 element was also allegedly found in Pfizer vaccine vials, which is an entirely separate, massive controversy going on right now. But let's stay focused on Louwen's claim about the Wuhan data. What exactly is an SV40 promoter mechanically?
Let's break down the acronym first. SV40 stands for simian virus 40. It's a virus that naturally infects monkeys, specifically macaques. Historically, it's famous because it accidentally contaminated early batches of the polio vaccine back in the 1950s and 60s because those vaccines were grown in monkey kidney cells.
Okay, Vix.
But decades later, molecular biologists studying this virus realized that a specific part of its genetic architecture, the promoter and enhancer region, was incredibly powerful.
What does the promoter do? Is it like a switch?
Think of a promoter as a structural landing pad for the machinery that reads DNA. When a cell wants to make a protein, an enzyme called RNA polymerase has to land in the DNA, read the gene, and transcribe it into RNA. The promoter is the sequence that screams, "Land here, start reading." The SV40 promoter is legendary in biology because it is an incredibly strong, aggressive promoter, especially when placed inside eukaryotic cells, cells that have a nucleus, like animal or human cells.
So, if a scientist wants a cell to pump out massive, unnatural amounts of a specific protein for an experiment, they take the gene for that protein and they staple the SV40 promoter right to the front of it. It basically tells the cell's machinery, ignore everything else, sit on this landing pad, and read this gene constantly, non-stop, at maximum volume.
That's a very accurate analogy. It is a genetic turbocharger used to force overproduction in a laboratory setting. Again, it is a specialized tool. It is absolutely not something you would ever expect to find naturally occurring, woven into the lung fluid of a human patient suffering from a novel coronavirus.
And then there was a third category of elements he claims to have found, which honestly, when I first heard it, sounded a bit like straight science fiction. Glowing proteins?
Yes.
Yes.
He claims to have found genetic sequences coding for GFP green fluorescent protein and RFP red fluorescent protein.
Let me guess, these are also standard tools you order out of a biology catalog.
Extremely common ones. GFP was originally discovered and isolated from a species of bioluminescent jellyfish. Scientists use the genes for these glowing proteins as visual markers, or biological tracking beacons.
How does that actually work in practice? Like, why make them glow?
Let's say you are studying a specific protein, and you want to know exactly where it travels inside a living cell. Does it go to the nucleus? Does it go to the cell wall? You take the gene for your protein and you attach the GFP gene to it. When the cell produces your protein, it produces it with the jellyfish protein attached. Then you put the cell under a specialized fluorescence microscope, you hit it with a specific wavelength of laser light, and that exact part of the cell literally glows bright green.
It's a literal highlighter pen for molecular biology. So let's just summarize the core explosive discovery Louwen is presenting here. He looks at the raw foundational genetic data from the very first patients in Wuhan, the absolute epicenter of the pandemic. And instead of just finding the natural genetic sequence of SARS-CoV-2 making its debut, he claims to find antibiotic resistance genes used to poison unwanted cells, viral turbochargers harvested from monkeys, and the genetic code for glowing jellyfish proteins.
It's a lot.
He is finding the entire standard issue toolbox of a genetic engineer mixed in with the lung fluid of dying patients.
That is the core assertion. He has the data set he has formally reported to the national health authorities in the Netherlands, demanding an explanation.
Here's where it gets really interesting, because if he had just stopped there, it would be a massive story about the origins of COVID. But the interview doesn't stop with Wuhan. Louwen drops a plot twist regarding this data that completely shifts the paradigm and changes the entire scope of the problem.
Right. If he had only found these elements in the 2019 Wuhan data, one might easily argue it was an isolated incident, an anomaly related specifically to the outbreak at the Wuhan Institute of Virology. But Louwen claims he didn't stop there. He expanded his search parameters.
He kept digging through the public databases, and he says he found these exact same artificial lab elements in samples taken from modern COVID patients in 2024. But it gets crazier. He went backward in time. He started pulling historical genetic samples dating from 2012 to 2018. He pulled genomic data from completely unrelated studies on colon cancer, studies on bladder cancer.
And crucially, he stresses that he looked at both the data from the sick patients in those cancer studies and the data from the healthy control groups that researchers use for baseline comparisons.
And he claims he found these lab elements everywhere, in data from years before COVID even existed, scattered across different diseases and across different populations.
Which is exactly why he is sounding the alarm with such urgency. The interview frames this discovery not just as debate about the origins of a single virus, but as a systemic, catastrophic crisis concerning the integrity of foundational medical data globally.
I think we need to connect this to the listener right now. Why should you, listening to this, care about some abstract public data sets stored on a server somewhere? You should care because public data sets are the bedrock upon which all modern medical research is built. If a pharmaceutical company wants to understand how a new targeted cancer drug works, or if a university wants to find a genetic marker for Alzheimer's, they don't start from scratch. They download massive amounts of genomic data from these exact public databases to run comparisons.
Right, they rely on that data being pristine.
If the data they are downloading is fundamentally flawed, if it is polluted with artificial noise, the cures built on that data might be entirely flawed.
It is the equivalent of trying to build a skyscraper on a foundation of sand, but you don't realize it's sand until you're on the 50th floor and the walls start cracking. If the baseline data that researchers use to compare a healthy human genome against a sick genome is secretly polluted with sequences of glowing jellyfish proteins and monkey viruses, the entire premise of the resulting diagnostic research is called into question.
So we arrive at the million-dollar question. If these glowing proteins and resistance cassettes are everywhere, scattered across a decade of public medical data, how on earth did they get there? The interview presents two stark, wildly different hypotheses to explain this phenomenon. We'll start with the one Louwen explicitly states he hopes is true, the contamination crisis.
This is hypothesis A, and to grasp it, it relies on understanding the physical, mechanical reality of how genetic sequencing is actually performed in modern laboratories. Flavio, the host of the broadcast, uses a very relatable domestic analogy here that works perfectly.
I love the bread machine analogy. It takes something entirely invisible and makes it incredibly easy to picture. Flavio says, imagine you are using a public, communal bread machine at a community center to make a strictly gluten-free loaf of bread because you have an allergy. But the person who used the machine right before you made a heavy, dense walnut loaf. If that machine wasn't cleaned with absolute microscopic perfection, if they just wiped it down quickly, traces of walnut dust are going to get left behind and baked directly into your supposedly safe, gluten-free loaf.
In the world of molecular biology, the bread machines are the high-throughput genetic sequencing machines, like the ones made by Illumina or Oxford Nanopore. These are incredibly expensive, complex pieces of equipment. Most individual researchers or smaller labs don't have their own dedicated machines for every single discrete project. They share them, they run batches.
So on Monday, researcher A might use the machine to sequence the results of an experiment where they were heavily using the neomycin resistance cassette and the SV40 promoter to modify some cells.
Exactly. They run their samples through the machine's flow cells, and then on Tuesday, researcher B comes in, loads up the raw lung fluid samples from a vital clinical trial, and runs the exact same machine. Louwen's hypothesis is that these sequencing machines globally might be systematically cross-contaminating samples.
Oh, wow.
Yeah, the residual genetic material the walnut traces from Monday's highly concentrated lab experiment are physically bleeding over into the digital readouts of Tuesday's patient samples. It's a phenomenon sometimes referred to as index hopping or barcode swapping.
So it's a massive systemic problem of laboratory hygiene on a global scale.
If this hypothesis is true, the implications for the scientific method are staggering. It means we cannot inherently trust the pristine nature of the readouts in public databases. How can a researcher accurately identify the true, subtle genetic cause of an autoimmune disease, or measure the microscopic effects of a new mRNA vaccine if the data pool is constantly polluted with generic, loud lab tools that act as static, drowning out the real signal?
Okay, I have to push back here, though. Let's play devil's advocate. If this is merely a cleaning issue, if researchers just need to develop better protocols to scrub their expensive lab equipment or use tighter digital filters, why is Louwen treating this like a global DEFCON 1 emergency? Why is he writing formal legal letters to the inspectorate and the GMO watchdogs? It sounds like a really annoying IT and maintenance problem, not a public health crisis.
Well, I wouldn't call it just an IT problem because genomic analysis is no longer just a theoretical academic exercise done in a vacuum. It is increasingly the foundation used to make real, life-and-death medical decisions. We are rapidly moving toward an era of personalized precision medicine where treatments are tailored to a patient's specific genetic makeup.
Okay, that makes sense.
If the baseline databases used to design those complex treatments are fundamentally noisy, it destabilizes modern diagnostic science entirely.
Give me a concrete example. How does this hurt a patient?
Imagine a scenario where the sequencing machine tells an oncologist this patient's tumor has a specific genetic marker. But what if that marker is actually just leftover lab trash from a previous run on the machine? The doctor, trusting the data, might put the patient on an incredibly aggressive, highly toxic chemotherapy regimen that they don't actually need and that won't work on their actual tumor.
Or conversely, the artificial noise from the contamination might obscure a real, faint genetic signal, and a patient misses out on a life-saving targeted therapy because the machine couldn't see the real mutation through the static of the jellyfish proteins.
Exactly. The integrity of the data is paramount. Louwen is arguing that if contamination is this widespread and unrecognized, the scientific community is essentially flying blind while believing they have perfect 20/20 vision. They are drawing maps of the human genome based on a dirty lens.
That is hypothesis A. The machines are dirty, it's bad, it means a lot of science is flawed, but it's a mechanical error, it's fixable. But then the interview pivots to hypothesis B, and this is the truly terrifying one. Because what if it isn't a dirty machine? What if those artificial elements, the resistance cassettes, the SV40 promoters, aren't static on the lens? What if they are actually inside the biological reality of the humans being sampled?
Which leads us to the second, far more ominous hypothesis: the presence of a circulating laboratory-manipulated virus that has integrated these tools into its structure. To get there, the interview delves deeply into the contentious debate between the lab leak theory versus the theory of natural zoonosis.
Let's talk about how the interview frames the origins of the virus, because they pull no punches. They spend a significant amount of time attacking the legitimacy of the World Health Organization's official investigation in Wuhan back in early 2021.
Yes. The WHO sent an international delegation of experts to Wuhan to investigate the origins of the outbreak. The video actually plays a clip of the investigators, including Marion Koopmans from Erasmus and Peter Daszak from the EcoHealth Alliance, walking around the Wuhan wet markets, looking at frozen meat stalls, searching for a natural animal source.
And Flavio is utterly, openly dismissive of this entire trip. He characterizes investigators like Daszak and Koopmans with a very brutal phrase. He calls them butchers inspecting their own meat.
It's a very specific, highly aggressive framing. The underlying implication is massive conflicts of interest. The argument is that individuals who were deeply involved in funding, advocating for, or collaborating on coronavirus research, specifically people with close professional ties to the Wuhan Institute of Virology, should absolutely not have been the ones chosen to lead the independent, objective investigation into whether a lab leak occurred at that very facility.
But the broadcast goes further, noting that Koopmans initially, publicly, called the lab leak theory a conspiracy theory. They show a clip of her on a Dutch news program called Zembla. The interviewer asks her if the mere presence of the Wuhan Institute of Virology, a massive biosafety lab, actively studying and collecting bat coronaviruses located right in the exact city where the novel coronavirus outbreak started, made her even slightly suspect a leak. And she basically laughs it off. She says she had no association with that idea.
Flavio finds that response intellectually absurd. He uses a great analogy. He says, if a gruesome murder happens right outside the gates of a maximum security prison housing serial killers, wouldn't the very first, most logical place you check be the prison roster to see if anyone escaped? The interview argues that dismissing the lab leak outright from day one was a profoundly unscientific, politically motivated stance.
And Louwen makes it extremely clear where he stands on the origin question. Based on his analysis of the genomic data, he leans heavily, almost entirely, toward the lab leak theory. He argues that the virus SARS-CoV-2 appeared in the human population already perfectly, seamlessly adapted to infect humans.
This is a foundational argument in the origin debate. Typically, when a virus makes a zoonotic spillover, meaning it jumps from an animal, like a bat, into a human, it is incredibly clumsy. The virus's machinery is optimized for bat cells, not human cells. It doesn't spread well at first. It usually takes time, and often multiple intermediate hosts like a civet cat or a pangolin, to mutate and slowly adapt to human biology through trial and error.
It's like a key that almost fits the lock, but you have to jiggle it and file down the teeth for a few generations of replication before it turns smoothly and unlocks the door.
And exactly. But Louwen argues that SARS-CoV-2 emerged like a precision-milled master key. He specifically cites the presence of something called a furin cleavage site within the virus's spike protein.
Let's do a micro deep dive here. What is a cleavage site, and why is the word furin so controversial?
Okay, picture the spike protein of the virus, the little crowns on the outside. In order for the virus to enter a human cell, that spike protein literally has to be cut or cleaved in a very specific place to activate it. Humans naturally produce an enzyme called furin, which acts like a pair of molecular scissors.
So a furin cleavage site is basically a dotted line on the virus that says, cut here specifically designed for human scissors.
Precisely. And SARS-CoV-2 possessed a perfectly optimized furin cleavage site that made it incredibly devastatingly efficient at entering human cells and spreading. The controversy is that this specific cleavage site was completely absent in its closest known natural bat relatives, finding it perfectly formed on day one of the outbreak without any intermediate host showing the evolutionary steps is, to Louwen, like finding a turbocharger perfectly installed on horse-drawn carriage. It implies deliberate engineering.
The interview bolsters Louwen's stance by bringing up an incredible historical parallel involving Luc Montagnier, the renowned French virologist who literally won the Nobel Prize for discovering HIV.
Yes, the video plays a fascinating clip of Montagnier on French television way back in April 2020. At a time when the entire global scientific establishment and media consensus was forcefully pushing the natural origin theory and shutting down debate, Montagnier boldly, publicly claimed that SARS-CoV-2 was the work of professionals, the product of molecular biologists.
Montagnier claimed that his team had analyzed the genome and found sequences in the virus that looked exactly like HIV elements added onto a coronavirus chassis. The host of the interview points out how Montagnier was treated. He was laughed out of the room in 2020. He was vilified in the media as a crank. But now, 4 years later, the interview claims the narrative is shifting, the evidence is mounting, and these early mocked warnings are being vindicated by granular data like the anomalies Louwen is tracking.
Which brings us to the core mechanism of how such an unnatural virus would actually be created. To understand Louwen's deepest, most existential fears about what hypothesis B truly means, we need to do a deep dive into what gain of function, or GO, actually entails.
Let's explain it simply. What is gain-of-function research?
In the field of virology, gain of function is the practice of artificially, deliberately modifying a pathogen in a laboratory to give it new abilities that gain in its function. This usually means intentionally mutating it to make it more transmissible between mammals, making it more lethal, or, crucially, altering a virus that only affects animals so that it gains the ability to infect human airway cells.
They are purposely upgrading the weapons and armor of the virus, and the interview explicitly connects this practice back to Louwen's own past work with those lentiviral vectors we discussed in the first section.
This is where the science gets truly alarming, and where the implications become permanent. Louwen explains that lentiviral vectors, the genetic delivery trucks, are derived from retroviruses. HIV is the most famous example of a retrovirus. The defining, terrifying characteristic of a retrovirus is how it handles its replication cycle. Unlike a normal cold virus, which just enters a cell, uses the cell's machinery to print copies of itself and then bursts out, a retrovirus goes much deeper. It takes its own genetic code and uses an enzyme to permanently splice it or integrate it directly into the host's own foundational DNA.
Let's build an analogy for this because the difference is profound. A normal virus is like a burglar who breaks into a printing press, prints 1 million flyers of his own face, and then burns the building down as he leaves. But a retrovirus is a saboteur. He sneaks into the printing press. He doesn't just print flyers. He goes to the massive metal type plates that the press uses to print the daily newspaper. And he chemically alters the metal plates themselves. He permanently rewrites the instruction manual of the printing press. So now, every single newspaper that press prints for the rest of its existence includes the saboteur's message.
That is a phenomenal analogy. The alteration is permanent and structural. In gene therapy, like Louwen was doing with the rats, that permanent integration is incredibly useful. You want the cure to be a permanent fixture in the DNA. But Louwen warns that these lentiviral vectors, these permanent saboteurs, are also widely used as common tools in gain-of-function research to modify other viruses. And you can literally order these vectors out of a catalog if you are a researcher.
So researchers are using genetic tools derived from retroviruses to modify things like coronaviruses. What is the specific danger Louwen is warning about?
The immense fear is recombination. Biology is messy. Nature is chaotic. If a researcher is working with a highly contagious respiratory coronavirus and simultaneously using lentiviral vectors in the same environment or the same animal models to modify it, there is a risk that the genetic material recombines. It shuffles together. You could theoretically accidentally create a live, replicating coronavirus that spreads easily through coughs and sneezes, but that now possesses the retroviral ability to permanently integrate its genetic code directly into the host's DNA.
Let me just sit with that and process what that actually means. You're talking about a highly contagious respiratory virus that you catch on the subway like a common cold, but instead of clearing it in 2 weeks, it behaves mechanically like HIV, permanently altering your genome.
That is the ultimate nightmare scenario Louwen presents under Hypothesis B. If such a lab-created chimera escapes, it doesn't just cause a temporary wave of acute illness, it literally becomes a permanent, inherited part of the human genetic lineage. He warns this could lead to an unimaginable, massive rise in immune-related diseases, chronic fatigue syndromes, or bizarre cancers decades down the line because our foundational DNA is being silently scrambled by an artificial pathogen we don't understand.
The interview explicitly connects the specific fear to a figure named Judy Mikovits. They mention her as a scientist who previously tried to warn the establishment that the global blood supply was contaminated with retroviruses stemming from careless laboratory research. And they note that she, too, was heavily punished, legally pursued, and discredited by the scientific establishment for speaking out. Regardless of what you think of Mikovits, the interview paints a picture of a recurring pattern: independent scientists finding lab-created dangers bleeding into the public domain, sounding the alarm, and being mercilessly silenced by the institutions responsible.
Which leads directly to a massive, glaring ethical paradox that hangs over this entire conversation. If manipulating these viruses is so inherently, existentially risky, if a single leak could literally alter the DNA of the human species permanently, why on earth is the scientific community doing it at all? Why are biosafety labs operating these gain-of-function experiments?
That is exactly what Flavio asks Louwen. He basically says, listen, it sounds to a layman like we are just building advanced bioweapons. Why is this legal? Why is this funded by taxpayers?
The stated justification for the scientific establishment for gain-of-function research is a concept called pandemic preparedness. The argument goes like this. Nature is a massive, relentless biological laboratory. It is constantly mutating viruses. Eventually, nature will randomly stumble upon the genetic combination that creates a supervirus capable of causing a devastating global pandemic. If we just sit back and wait for nature to do it, we will be caught completely off guard and millions will die while we try to develop a vaccine from scratch.
So the logic is essentially preemptive strike. We need to build the supervirus ourselves first in a highly controlled laboratory so we can study its weak points, understand its mechanics, and develop a working vaccine for it before nature ever invents it.
That is the dual-use nature of the research. It is classified as defensive, preventative research, but the actual mechanics of it look exactly identical to offensive biological weapons development. The interview highlights a very famous, incredibly controversial Dutch example to illustrate this extreme risk. The work of Professor Ron Faucier, who, not coincidentally, also worked at the Erasmus MC.
What exactly did Faucier do that was so controversial?
Several years ago, Faucier was working with the H5N1 avian influenza virus, bird flu. Now, H5N1 is highly lethal to humans who catch it directly from birds. It has a terrifying mortality rate. But historically, it doesn't spread well from human to human. It lacks the specific mutations to be airborne transmissible between mammals. Faucier decided to change that. He deliberately mutated the H5N1 virus in his lab, and he repeatedly passaged it through ferrets.
What ferrets?
Because ferrets are the gold standard animal model for human respiratory transmission. If a flu virus can spread through the air between ferrets in separate cages, it is almost certainly capable of spreading through the air between humans. Faucier kept mutating and transferring the virus until he achieved his goal. He created an airborne, highly transmissible version of one of the deadliest flu viruses on Earth.
He essentially, intentionally, built the exact apocalyptic pathogen we are all terrified of.
Yes, he proved it was biologically possible. But the crisis occurred when he tried to publish his methodology in a scientific journal. He wanted to publish the exact step-by-step genetic recipe for how he created this potential doomsday virus. It triggered a massive, unprecedented international security crisis. Scientists, intelligence agencies, and biosecurity experts debated furiously whether publishing the paper was literally handing a blueprint for a global bioweapon to any terrorist organization with a decent laboratory.
It feels like a massive, agonizing paradox. We are creating the monster in the lab so we can practice fighting it. But the greatest, most immediate risk of the monster attacking the village is if it escapes our own carefully constructed cage. We are taking the theoretical, distant risk of a future natural pandemic and actualizing it into a physical reality right now just to study it.
And Louwen's data, If his second hypothesis about the artificial elements being inside the patients is correct, strongly suggests that the monster did, in fact, escape the cage.
So we have looked at the data Louwen claims to have found, the neomycin cassettes, the SV40 promoters, the glowing jellyfish markers acting as artificial barcodes in the earliest patients. We have unpacked the mechanics behind the two terrifying hypotheses: a global crisis of contaminated sequencing machines or a circulating DNA-altering lab virus. The interview has laid all this complex science out. So what is the Lighthouse TV broadcast actually asking for now? Where does this go? What is the fallout?
The interview culminates in a very specific call to action. Louwen is making it clear he's not just sitting on this data making YouTube videos, he has formalized his concerns legally. He states that he has formally notified several key Dutch regulatory authorities. The RIVM, which is the National Institute for Public Health and the Environment, the Health and Youth Care Inspectorate, the IG, and various specific legal watchdogs responsible for overseeing the safe use of genetically modified organisms.
He is drawing a line in the sand. He is demanding that they do their jobs. He is saying, I have handed you a data set that provides evidence of either massive, systemic data contamination that renders a decade of our public health science unreliable, or it provides evidence of an illegal, circulating man-made laboratory creation. You have a legal statutory duty to investigate this anomaly.
He is directly challenging the establishment. He is daring them to either prove him wrong by rigorously, transparently explaining the anomalies with alternative science, or to admit that a catastrophic failure of safety has occurred. He wants other independent, unaffiliated bioinformaticians to run his exact analysis on their own downloaded datasets to see if they find the same glowing proteins and resistance cassettes hiding in the code. He wants replication.
And as we evaluate this source, we also need to look at the broader media context surrounding this broadcast. This interview between Flavio and Louwen doesn't exist in a vacuum. It is part of a much larger, growing ecosystem of alternative media that is aggressively challenging the mainstream medical narrative.
Yes, the context is very revealing. Towards the end, Flavio specifically mentions an upcoming premiere of a documentary film called De Stilte Schroot, which translates to The Silent Screams. He notes that the premiere is being held at the highly prestigious Pathé Tuschinski Theater in Amsterdam, and it's directed by Frank de Rooij.
And the broadcast tells us exactly what the thematic focus of this film is. It deals heavily with unexplainable excess mortality, the statistical reality that more people are dying across various age groups than historically expected, and it focuses intensely on claims of widespread vaccine damage. Flavio mentions they will have a live panel discussion featuring figures like Andreas Voss to publicly debate these issues.
Additionally, if you watch the video, the broadcast visually displays its sponsors throughout the interview, featuring companies like Pro-Privacy, Bundle ITAR, and various alternative supplement companies. This context is important to note impartially because it paints a very clear picture of the media landscape this information is emerging from. It is an ecosystem that fundamentally distrusts centralized medical authority, prioritizes individual digital and bodily privacy, and actively seeks out evidence of institutional malfeasance and cover-ups.
So what does this all mean for you? Why have we spent the last hour meticulously dissecting a Dutch molecular biologist's claims about microscopic data anomalies, resistance cassettes, and viral promoters?
If we connect this to the bigger picture, it means that the foundational data of molecular biology, the very genetic code that dictates our understanding of human disease and drives the development of our future cures, is currently the subject of a bitter, high-stakes, and intensely polarized dispute.
It means that the established scientific consensus is being actively, aggressively challenged from the outside by highly credentialed people who used to be on the inside. Whether Dr. Louwen has simply uncovered a massive, embarrassing issue of sloppy laboratory hygiene with those expensive sequencing machines, or whether he has legitimately stumbled upon the suppressed genetic fingerprints of a lab leak that permanently changed the human genome, the basic integrity of the data guiding our global health policies is under intense fire.
We have summarized the two paths laid out by Dr. Louwen. On one hand, we face the prospect of a scientific community that is blindly trusting contaminated machines, essentially building multi-billion-dollar medical houses on sand. On the other hand, we face the terrifying reality of a world dealing with the permanent genetic fallout of an escaped, engineered laboratory experiment with synthetic viral elements potentially integrating into human biology forever.
As we promised at the beginning of this deep dive, we aren't here to tell you which of those paths is the absolute truth. The polarization around these specific claims is extreme, and the science is complex. But the debate itself is undeniably crucial for you to understand because it dictates the future of medicine.
It forces all of us to ask critical, uncomfortable questions about data transparency, about the extreme risks we are willing to accept in the name of scientific progress, and about how much blind faith we place in the institutions that manage those existential risks.
I want to leave you with a thought to mull over. Think about the timeline of human history. For hundreds of thousands of years, nature wrote our genetic code, and we just lived it. We were passive passengers on the ride of evolution. Now, in just the last few decades, we have acquired the profound tools to actively edit the book of life. We have CRISPR, we have lentiviral vectors, we have gain of function, we can write our own code. But if Dr. Louwen's contamination theory is right, we might be scribbling haphazardly all over the pages without even realizing it. Are we technologically advanced enough to edit life, but not wise enough to clean the pen? Keep asking the hard questions. Keep looking at the data from multiple angles, and never stop being insanely curious. We'll see you on the next deep dive.